Okay, let's get started. So good afternoon, everyone. Welcome to the GCC single cell omics panel discussion. So before we start, just a few housekeeping notes. So the talks will be recorded. So you don't need to be busy, take screen capture or take notes. So it will be recorded and posted on the GCC website. You can always check it out. And then for the so there will be three of us do a very brief presentation, myself and Nick and then Zhongming. Each only will take about 10 minutes. And so therefore we'll leave about 20-30 minutes for Q&A. So if you do have a question, you can you can type it into the Q&A box. And if you can specify which people you want to direct your question to, that'd be great. That will help us to answer the question. So this is one of the. This is really the first panel discussion series. Is focused on introduction, introduce single cell technology in the lab, local resources. So this is one of the many to come. We hope. We already, as you see, we pick up quite a few topics. Those are not like finalized, but for example, there will be one focused on data analysis and immunology and cancer research, multi-omics, so on and so forth. This is one of the effort that our GCC single cell cluster try to put together for education purpose. So therefore, the topics actually should be driven by people like you. So if you have any thoughts and feedback, feel free to reach out to us and to GCC, say which topics you would like to see. We can organize sort of panel discussion. This is really critical for the series to run successfully. As I mentioned, we plan to have a large proportion of the series allocated to Q&A. So this is quite different from other topics, like a seminar. You see, you have been sitting through so many times. I guess this will be more education or introductory or specific topics. So that's why we feel like we want to leave enough time for interaction. And right now we only can do online, but in the future, hopefully we can do it in person. We are planning to be a bi-monthly or two, 3 months. We'll have one panel push it up. We'll give you more announcement when it's getting close to it. So for today, I will talk to you quickly about GCC single cell omics cluster first, if you haven't heard about it, and then I will introduce a few, the RNA and the ATAC-Sea, the most commonly used single cell technology these days, and then also the resource at Baylor, and then we'll be followed by Nick, will introduce about DNA and then cancer, and also research at M. Dennison, and finally Zhongming Zhao from UT. We'll talk about a little bit on the data analysis and then the resource at UT. So the GCC single cell omics cluster has been formed for about 1.5 years now, and then it currently have all 7 GCC institutions as part of the cluster. It's chaired by myself and co-chaired by Nick Nevin and Louis at Rice. And we have 14 committee members, 2 from each institution. So if you have questions or comments on how the cluster can better serve you, feel free to reach out to the committee at your institution. Their name is on the GCC website. So we currently have more than 150 faculties listed as a member. So the reason we have GCC single cell omics is because the single cell technology, as you know, is an emerging technology. It's really multidisciplinary and it's really new and actually rapidly evolving. And it really can be used by many different labs and disciplines and field. And then there are significant barriers, including experimental design, process, and also as well as data analysis as well. So that's why that's what motivates us to form the GCC omics cluster. The idea we have this one is really, as I said, is education and training. We would like to provide, a sort of more coordinated effort, to better educate and help you to embrace this new technology, and also we are trying to how to say, not to centralize, but to have a place where you can find all the services locally, so you can match your need with the labs. And finally, we want to promote research collaborations among people who either developer or user of the technology, and for for their research. So to achieve that, we have weekly seminar for education purpose that run by Nick Naven and MD Anderson. So the plan is it used to be one weekly seminar, but because of COVID and it's got stopped. I think it will be restart early next year. And we also, as a cluster, we host workshop, and then the panel discussion as we kick off the day. We have summer one hand workshop on camp, planned. Once we can do it in person, there will be both experiment and dry lab camp. And once it's how to say possible, then we'll give you announcement, and then people can sign on to it. And of course, we have the annual symposium. The last one was held a couple months ago, and then we'll have another one coming, hopefully in person, during fall this year. So there's also quite a few core lab locally provide single cell technology services, Baylor and Janssen, and then also the UT. Annually research collaboration, the cluster has been involved and organized interaction between single cell groups with many other groups, for example, mental health or pain, things like that. And then hopefully we can align the technology people versus the or more biological focused labs. And then also we are actually help to people to connect, so to respond some of the funding opportunities. So the goal really just try to beef up the activity here in at the TMC, so we could be a leading position in single cell omics field. So let me move on to the technology. I think for most of you should probably very familiar with the single cell technology to a certain extent. So the main motivation in my mind is to overcome the barrier of the cell heterogeneity in the tissue. So you instead of taking the average measurement, you look at the cell type or individual level. And also it's really a good efficient way to find the rare cells population in your tissue, because which is basically missed by previous bulk sequencing approach. And finally, I think it's emerging, more and more people are using it, is the high throughput screen, like combined with CRISPR and other things, and then single cell to do high throughput screen. So the most popular technology right now, I think I would say, is the RNA single cell RNA-seq. The first paper, this technology is developed almost like 2012 paper in this Nature Technology paper, is basically use the SMART-seq, what they call SMART-seq, and those are done individual in the tubes. Basically, the method is you lyse the cell, you put the oligo DT primer with some universal primer, and you do the RT. And then the nice thing, the interesting thing part of it is called template switch. Once your RT synthesized around to the end of this, then you have a template independent adding of Cs, and then that will complement with what we call TSO template switch oligos. So that allow you to do PCR and amplify. After you have the full cDNA, and then you can do tegmentation or other method, to share them to small pieces. You can sequence. So, and then this single cell sequencing RNA sequencing technology is combined with microfluidic system, whether it's well-based or channel-based or droplet. I think really the field take off because of like Carol's group at publishing the work they published in Cell in 2015, where they really combined the single cell with the bead and droplet based. This is schematic. Basically, is by make the oil drop, make the oil droplet. You can have each droplet. Within each droplet, you can put a cell and the beads, and the beads carry each beads are uniquely barcoded. So with within a droplet, the cell are lysed, and then the RT which I showed before, the SPARSIC type thing, happened the RT expression, RT experiment, RT amplification, and then since there's a bit, each bit have a unique barcode, then all the cDNA synthesized in a gel bit have a unique barcode that specify which cell it come from. So after you done the RT, and then you basically break the oil droplet, and then that's become like a small bulk experiment, and you can make a fragment, and then you can basically sequence the outcome, because the cDNA have the barcode on it, so that you can then informatically go bin all the sequence with the same barcode into the same cell. So that's basically how it works. So what you can use this for, the most commonly used is looking for different cell types, in the same tissue based on their transcriptome. So this is one of the data set generated by our lab. Is we're looking for human retina cell types. There's about 60, 70 different subtypes, and then as you can see, this is a UMAP where based on the single cell RNA-seq, you can cluster them into different cluster. Each cluster will be corresponding basically to each different cell types. And then the bottom UMAP is a geneless. You can see genes have a very distinct. The dark part is the gene have very distinct pattern only in one cell type. Those are basically cell markers. It's more clear. You typically see the violin plot. You can see for each cell types, their genes are highly expressed, and you specifically expressed. Based on those gene markers, you can identify the cell types. Of course, some of the unique expressed genes also have very unique cell type specific expression pattern. So this is just a few examples. Another way to look at the other violin plot. The blue color is the highly expressed the genes, and then you can see PTE6A, which is the photosensitive cells, highlight the blob to your right, and the OnCAR2 is horizontal cells, highlight the one at the bottom left. So and also, as I said, this is really effective in finding rare genes. So in this data set, we are able to find RGCs, which is basically these small green clusters are basically opportunity for expressed RGC cells. In the retina, it really only count for 0.1% of the cell population. So when it is possible to get those really, really rare cell types. Example application, cell type specific expression, identify cell type specific differentially expressed genes, right? And you can also do linear tracing if you look at multiple time points, or sometimes you also put a barcode in the cell. You can look at cell linear tracing for progressive change over time, whether it's disease, whether it's development, whether it's trait to response to some job. And also, I mentioned early, the transcription or transcriptome can be as a phenotype readout that you can use for high throughput functional assay or screen. So in addition to transcriptome, in the last year or so, the single cell ATAC also become more and more popular. So this is really a technology basically combine the ATAC-Sea with the single cell technology. So the ATAC-Sea, the principle of that is looking for basically open chromatin. The reason is like when you have a chromatin, the more open chromatin part, if you incubate it with transpose element, it can be more easily inserted into the open chromatin region. And in the transpose element, they have a adapter sequence assembled in there. So when you have two next to each other, then you can PCR out the region that transpose element insert in. So when you sequence the fragment, then you can form based on this distribution, you can basically identify regions that is open. So those open chromatin are basically corresponding to promoter, enhancers, but also CTCF binding part and things like that, but typically just more accessible to transpose element. So when you combine this to the microdroplet system, you can also generate, based on the chromatin profile, different cell types. So this is again human retina. You can see, based on the chromatin profile, you get a similar sort of cluster type plot, and then each one corresponding to different cell types. So this is just quick look at some of the raw data. And the up two lines are bulk retina. You take the whole retina, you do the ATAC-Sea, and you can see some peaks. And the left one is a rod specific gene PD6A, and you can see green and the red peaks is the promoter region. The red one, the red one is the promoter region. The green one are enhancers. You can see some above a little bit things, but when you look at the cell type like rods, you can see much more from signal in those green region. And this is even more obvious when you look at the rare cell types. If you look at the SLC6A1 genes that is highly expressed in amacrine cell and the mural glial cells, you can clearly see there's much more signal in the mural glial cell and AC, but really barely nothing in the bulk. So this will allow you to looking at relatively rare cells, the chromatin region. And of course, once you have RNA and ATAC, you can combine them together. Then you can get both the transcriptome and chromatin profile for that cell type. So this is just sort of align the single cell ATAC and the RNA together. In one of the cell type, we're looking at either one of the data set, and you can see these two data can be embedded or aligned properly, and then we'll probably talk more about it, and then you can get the profile. And then this can be done informatically. There's a software by SC. We tried quite a few different ones, but this one developed by Ken Chan's group at MD Anderson seemed to perform really, really well. And they just submit the paper, and then the software is downloaded. Of course, the integrated RNA and the ATAC is informatically done. If you have the data are generated independently. But now, just recently, the 10X also just released a new kit called multi-omics kit, where you can profile both RNA and the ATAC in the very same cell at the same time. So then you don't need to have the guesswork on which one is matched to which. So this is just a quick snapshot from the Loop browser, where you can look at the different cluster, and then you can see the green, the blue one is the promoter, and then there's a co-accessibility, meaning that the other peaks have similar profile with the promoter if they go up together and go down together. And then also you can look at different cluster, the gene expression, and see which cluster corresponds to what. So basically look at you really can combine these two data very seamlessly together. So as Baylor single cell core facility, I'm the director of it. We offer fair amount of different single cell services. So this also first we really help you to work with you on training and consultation of your project and design project. We offer single cell RNA-seq and ATAC-Seq, and as I mentioned, multi-omics. I already tested out in the core. We actually formally offer that service, and then also for immunology folks, we also offer the total seq, meaning look at the protein surface marker and RNA at the same time. And we're currently working on the single cell histone modification profiling that's coming soon. So if you're interested in Baylor core, you can go to the ATAC-Sea single cell genomic SCGC. That is the link, and you can see more information on that. I think that's it from me. And then Nick, do you want to? Let me stop sharing.
Sure, I'll take over from here. Thanks. Okay, well, I hope everyone's having a good afternoon. I'm going to just take about 10 minutes or so to talk about single cell genomics and cancer research, and also talk a little bit about our own CPRIT Single Cell Genomics Center. Oops, sorry, 1 second here. Just change something. All right. So as Ray mentioned, it's the field is growing tremendously. I did a PubMed search recently, and there are thousands of articles now. I think by towards the end of 2020 on single cell sequencing. A while ago, we wrote a review article, and we kind of try to classify the publications that were out at the time, although it's a little bit outdated, but you can see that areas like cancer research really have probably the most publications now. This is increasing, but areas like normal development, microbiology, neurobiology, immunology are also have a lot of single cell sequencing applications. And there's still quite a lot of papers, of course, that are on technologies and computational tool development, just because these areas are still maturing. Our own work is focused a lot on single cell DNA sequencing. When I was a graduate student at Cold Spring Harbor, we developed one of the first single cell DNA sequencing methods to look at copy number in tumor cells. Since then, we've developed methods to do kind of whole genome sequencing to look at mutations at base pair resolution, so both single cell exome sequencing and whole genome sequencing, which we've published in a few papers now. This turns out to be very expensive because if you sequence just one cell, it's the cost of a human exome or genome, which is around $1,000 or $2,000. So a lot of our efforts following that focus more on targeted areas so that you can profile lots and lots of cells, but then focus a little bit more on smaller spaces of the genome to look at mutations. Methods like single nucleus targeted DNA sequencing. There are also commercial platforms like Mission Bio that do this now. And then more recently, we've developed a lot of tagmentation-based methods to look at copy number that are higher throughput, like acoustic cell tagmentation. So yeah, so in terms of the actual technologies that exist, Ray covered a lot of these, so I'm just going to briefly mention them. Single cell RNA sequencing is definitely the most mature. It's an area that I think a lot of people work on, not just the cancer field or any specific field. And so, you know, right now, the gold standard platform, I would say, is the 10x Genomics 3' single cell RNA sequencing. You can look at thousands of cells, quality is very good, lots of genes per cell. Single cell DNA sequencing, there are, you know, a bunch of technologies. None of them are, I would say, the most widely used. And so there's methods to look at both mutations, either plate-based or fax-based, like ones that we've developed. There are commercial systems like Mission Bio to look at mutations. You can look at around 1,000 amplicons now that target about 1,000 mutations across 5,000 to 10,000 cells. There are methods to look at single cell copy number developed by our lab, also by Saurabh Shah's group and others. And there's even a 10x Genomics platform to look at copy number, but unfortunately this has been discontinued due to some IP issues, and I guess they weren't profiting enough from this. So right now, if you want to do high throughput, you basically need to use, or we would recommend use one of the tagmentation-based approaches. And then single cell epigenomics. While single cell ATAC-Sea really has been commercialized, is used widely, methods for methylation and high-C and others are a little bit farther behind, and they still have a lot of technical errors. So most people doing epigenetics are mostly kind of single cell ATAC-Sea focused. And there's some higher throughput methods, just put them down here, called combinatorial indexing. This is mostly out of Jay Shender's lab at UW, and it's very, very scalable so that you can look at hundreds of thousands of cells and not tens of thousands. So one thing to think about, of course, if you're considering to do single cell sequencing experiments is that certain tissues are compatible with certain assays, and you can't do single cell RNA sequencing on all tissue types. If you're working with FFPE materials, for example, you can only really do single cell DNA sequencing. It's very difficult to do RNA or even ATAC. If you have SNAP frozen tissues in OCT or liquid nitrogen, you can make nuclear suspensions, and you can use that for single cell DNA sequencing. You can also use it for single nucleus RNA sequencing, which is not the, you don't get the kind of cellular messenger RNAs, but you get the nuclear RNAs. And you can also do single nucleus ATAC sequencing, although the quality is a little bit less than if you're working with cells. Now, if you have cells, either fresh viable cells, or if you cryostore them in freezing media like DMSO, you can basically run any of these assays. And so that's really the best thing to do, is to dissociate your tissue and then store it as viable cell suspension so that you can use it later for these single cell experiments. And if you're working with surgical samples or things from the clinic, you really have to think about fast processing protocols, and so that you can quickly get a sample after surgery, dissociate it, count viable cells, and then either cryostore it or enrich the cells, and then run the assays right away, or I'll store them and run them later on. So in cancer research, a lot of the early papers focused mostly on primary tumors and looking either at clonal evolution and mutations that occurred at the primary tumor site, including a few papers from our own highlighted here, or looking at the microenvironment. This is mostly by single-cell RNA sequencing by groups like Aviv Berjev and Martin Suva, Dana Pear, and others. And more recently, people focus a little bit more on circulating tumor cells, as well as looking at metastatic lesions. Work by Dan Haber's group, from Matthew Myerson's lab and others, our group as well. And probably the two new areas that I think are very exciting that us and others are working on include pre-malignancies, to look at early stage cancers where you're limited really in the number of cells you can analyze, but there's a lot of information that can be gained there, both from the microenvironment and from the tumor cell populations. And then really looking at pre and post-treatment samples, which has all sorts of other challenges in that you're limited by the core biopsy samples, or usually small samples that are taken before and after treatment. So what can you do with single cell DNA sequencing methods? One thing you can do is, whether it's mutations or copy number, you can take all your cells, cluster it in high dimensional space, and see how many subpopulations exist, and then look at what genomic alterations those subpopulations have. You can then take that data and try to reconstruct chronology and lineages to understand what are the order of events that occurred and what's the relationship between those subpopulations if you're looking at a genetic lineage. You can also look at how clonal dynamics change over time, so how the frequencies of clones change in primary metastatic tumors, pre or post treatment, or in pre-malignancies and invasive cancers. And then finally, this is one that's of big interest to the clinical community, is looking at mutational co-occurrence. So if you're looking at, say, 1,000 cells, how many of those cells are sharing, for example, these sets of mutations, and how many are mutually exclusive? So here showing you, a set of mutations that are mutually exclusive, like P10, CHD1, and they can see these are all mutually exclusive in clone 3, and that's important for targeted therapy. And then, of course, the tumor cells are not evolving on their own, they're co-evolving with the microenvironment. And so another big question is, how do the immune cells and the stroma change as tumors progress and become resistant to therapy? And especially things like fibroblasts, the vasculature, adipocytes, macrophages, and the immune system. And so with single cell RNA sequencing methods, you can do unbiased sequencing of a large number of cells. You can cluster that data and identify groups of cells that share common transcriptome profiles. You can then take that data, say just from the T cells, recluster it, and then identify different cell states. And those cell states correspond to different biological functions. In some cell types like T cells, they might be pretty well established, the CD4, CD8 cytotoxic lineages. But in other cell types like fibroblasts, those are not quite as well understood. And so you get this unbiased classification that's not marker dependent. You can also take the tumor cells and you can recluster the data and do a signature analysis to look at how different properties of tumor cells vary in the population, like hypoxia or EMT, invasion, migration, and so forth. You can also look at the coexistence of different subtypes that are usually defined by bulk RNA sequencing methods. Oops, sorry. And you can also reconstruct lineages to understand if there's a differentiation trajectory that's still preserved in the tumor. Of course, you can't do that with many cancer types, but there are certain cancers where you can. And then, as Ray mentioned before as well, you can do cell type specific differential expression analysis to understand how are fibroblasts, for example, different pre and post treatment, and really just focused on those cell types. There are also many translational applications that we and others are excited about. Looking at early detection in fluids, can you look at single cells that might be in the urine for bladder cancer or in the sputum for lung cancer? Can you do risk stratification for patients, predict which ones will progress or be resistant to therapy? Can you improve matching to clinical trials or predict therapeutic response? And then there's a lot that you can do with discovering new drug targets. So can you identify new drug targets in fibroblasts and tumors, for example? And non-invasive monitoring, so that's in the blood, looking at CTCs and understand if you can track disease progression and resistance from liquid biopsy samples. And then also think about lineages, which mutations you really want to target. Do you want to target truncal mutations that are inherited and present in all cells, or do you want to figure out what's the most malignant population and just target that? So let me tell you a little bit about our single cell sequencing core. We're funded by a CPRIT grant, and so we're more of a collaborative center for doing projects. And here's some members of the core, especially want to mention Tuan Tran, who's leading our core, and Jia Hong, as well as some data scientists like Jerome Lin, Min Hu, and other people from our lab like Emi and Shan Shan, and also Kyler, who leads a lot of the technology development. So our single core has three parts to it. It has both a tissue dissociation facility, because the rule of thumb in genomics is garbage in, garbage out. And so you want to have the highest quality samples going into your experiments. The single cell technologies, both commercial platforms as well as new things that we're developing. And then finally, the data analytics, so that we can really understand these large scale data sets. And there's also sequencing that's performed, but that's not part of our core. We actually do that in the core adjacent to us, which is a sequencing core facility. So the tissue dissociation is very important, especially for RNA assays. And so we have a facility set up where we can take incoming samples from surgeries, from cores, from biopsies, from every possible type of tissue that you can imagine. And then we try to develop protocols for different tissues, like because the breast is very different than the liver or the lung or the kidney. And this is all fairly open source. So most of these protocols we post on a protocols.io. We've done a lot of work on breast tissues, for example, but also other tissue types. Part two are the technologies and assays. So this is currently what we have available in our core. For single cell DNA sequencing, we have the Mission Bio system. We also have our own assays we developed for copy number, both plate-based and also nanowell-based on this Wafergen system, where we have a customized protocol that we implemented. We have automation on this acoustic liquid transfer system called the Echo 550. And then single cell RNA sequencing, that's kind of our workhorse. So we both the 5' and 3' technologies, immune profiling also on 10x, and then the single cell ATAC-Sea also on 10x, and the co-assays as well, which we're just starting to use, are available. And we also have more specialized platforms that we use too for various projects. We have a FACS system to enrich or isolate very small cell populations, or separate immune stroma tumor cells. We have LCM systems so that we can cut out individual cells and tissue sections and isolate them for analysis, which of course is more technically challenging, but in some cases it's very useful, like in pre-malignant cancers. We have CTC isolation platforms like the RareSite. We also have live cell imaging and micromanipulation platforms like the ALS system. And then a big part of our CPRIT Center is that we also have a lot of data processing and analysis support for these projects, both on the single cell DNA analysis side, single cell RNA analysis, and single cell ATAC. And so we're fairly experienced in these different areas, and really most projects that we take on in our kind of collaborative center, we try to see through from the beginning all the way to the end. And so we'll often work with investigators for quite a while on their data analysis parts of the project. And then the other activities, Ray mentioned a few of these. I just wanted to put them up here again. If you're interested, we have a single cell seminar series where we invite people from the TMC to give talks. This was on a weekly basis pre-pandemic, and after the pandemic, we shut down for this fall semester. We're going to be starting it again this spring. We also have 10K user grants, and so these are to help support small projects and get them off the ground, pilot projects. We're going to be organizing a single cell genomics workshop. We're going to wait a little bit after the pandemic, I think, to organize this because it's intended to be in person, although we might organize a kind of data analysis workshop prior to that. And then we provide a lot of user training. So a lot of people just want to know how to dissociate tissues, how to learn more about 10x, all those things. And you can contact us and reach out to us, and we can try to help you with whatever project you're thinking about doing. It's all in Agilent and ilabs, so quite convenient if you're trying to make service requests and set up projects after kind of meeting with us and discussing the project. And I think that's it. Just wanted to mention again all the people that are involved in our core. That's where it's located. And if you have a project and you think would be interesting to collaborate on, feel free to reach out to us and we can set up a meeting and talk about it in more detail. That's all I have. I think we're saving questions to the end, so I will stop sharing my screen. Okay. So I think Zhangming is going to give the next talk, correct?
Go ahead.
Yes, I'm connecting. Okay, I think you can see my screen, right?
Yes. Yeah.
Okay, that's good. Yeah, so I'm the last speaker today to introduce sequencing, especially today single cell sequencing facility and what we do in UT Health. Actually, I did the opposite way. I didn't realize. I have the first slide, second slide to introduce the CPRIT cancer genomics core, and then I will introduce a few projects briefly, what we are doing, what we can help other people. So, and my name is Zhongming Zhao. I'm the director of this cancer genomics core, founded by CPRIT. This is a website, and if you are interested in later on, you can visit the website. It includes all the service and the price information there. And the core is located in UT Medical School building, which is convenient to many investigators. So this is actually not very updated. The summary of the core currently we have. So my background is mainly in genomics bioinformatics. So actually we do a lot of those kind of data sequencing data analysis, and also the core we had the Illumina sequencing machine and also 10x genomics controller, so we can do the regular sequencing plus the single cell sequencing. So the team has collaborated with many investigators in TMC area. Also, my track record before, we worked with many other investigators in the genomics data analysis, leading to many productive research projects. So here, these slides, I like to briefly summarize what we currently do, what we have, using a few examples here. The first one is like, Ray and Nick mentioned, there are many kinds of single cell sequencing techniques in addition to the traditional, RNA sequencing, which is now becomes more and more popular. So my core recently also developed single nucleus RNA sequencing. So this is really promising because we have many frozen tissue or FFP samples. So my second part here is mainly about two methods we developed in the core. The first one is mainly to address the batch effect using this kind of triple neural network, the deep learning approach to integrate. Like currently we can do millions of single cell sales data, and using even desktop to do this kind of batch effect, adjustment. And the right side of the visualization actually showing how it works. And later I may have one or two slides, depends on time. And the second one is the driver. Today I will briefly introduce this. The driver means it's a program, software program. Also underlying is a deep learning method. We use this program, the method to basically to identify those kind of important critical transcriptional program regulator, mainly like transcription factor from single cell RNA sequencing data. So this, I will introduce this method a little bit later. And my third part of this slide is mainly data analysis. Like my lab itself, we recently analyzed the lung tissue single cell RNA sequencing data came from the those kind of COVID-19 patients, from the mild to severe, those kind of symptoms. And we found a few gene signatures, especially the open reading frame 10, which is the expression level can be indicative of the severity of the COVID-19 syndrome. And also we did those kind of, I involved in a project for those kind of lung injury, and then to see how it recovered as a more like a trajectory, the time series, and using single cell RNA sequencing. The right side, the bottom figure showing this kind of the trajectory called, or a software tool called RNA velocity, showing those gene expression change during the lung recovery after the injury, and eventually it merges to a cell type called KRT8+, those kind of cells, gene expressions labeled cells. So that's another example. I don't have time, just showing you this kind of single cell sequencing can help us to investigate the dynamic change during the cellular system, and also can be in the response to the external like environmental changes. So my lab actually a few years ago published one of the first single cell RNA sequencing database And right now we just don't think it's impossible. It's impossible to maintain it. And also today I use another example called the CSEA, cell type specific enrichment analysis of the genetic variation, and we develop a database to illustrate how such kind of data can be used for the genetic study. So this is one of the examples I would like to briefly mention today, called the DrivAER. The full name is the identification of driving transcriptional programs in single-cell RNA sequencing data. This work was mainly developed and was done by Lucas Simon, who was a junior faculty in my lab. And the paper actually was officially accepted last week, published last week after long review process. So this slide showing how this works. Basically, you have a single cell RNA sequencing. Then basically we use a dimension reduction, and this is a method called a deep counter AutoEncoder. It's a deep learning method developed by Lucas Simon before he joined my lab. And then those single cell sequencing, the gene expression can get those kind of more like a dimensional, two-dimensional representation. And then we use a random forest, a classical machine learning technique to do those kind of feature analysis, try to find those genes which showing the different, from the different cluster, showing the enrichment of the gene expression. And then Lucas basically proposed this called a relevance score. Relevance means for each cell cluster, those kind of functional importance mainly focus on the transcription factor. So then this score, the higher score means more likely functional importance in the specific cell types. And the bottom two figures showing this, showing one example. Actually, he used two blood-related single cell RNA sequencing. So this is one of the two data sets. It's a monocyte, and those kind of top rank transcription program, mainly the gene, the biological pathway. And the first one, we use this to show like interferon gamma response has the highest, the relevance score that we call the I score. And this right side showing this distribution, the heat map showing these two group of cells by this specific marker, the marker set under the gene expression change, which is showing very distinct gene expression features from single cell RNA sequencing data. And this is, I don't think I have time, just very briefly, the insight, integrating millions of single cell using, this called a batch-aware triple neural networks. And we show this kind of, dynamics here. One, my former postdoc actually did this visualization. So using this triple neural network, we were able to show it has power to distinguish, to separate the cells by cluster from multiple batches. And also the second one is the scalability. For a few millions of cells using a desktop, we can make this kind of work done in like 25, 30 minutes. So this is a more like a conceptual view. Initially we have 3 batches labeled by different colors, but really our goal is try to see those gene expression across cell type, and the batch is very strong here. And after those triple neural network machine learning, eventually we were able to demonstrate the gene expressed across the cell type cluster, not by batch. And I hope I have a few more minutes to talk about another case study. We mainly focus on the genetics, and in genetics, like a genome-wide assertion study, we have many genes, and those genes we investigate their genetic variation, like a mutation, DNA label, and to see how they associate with the specific disease or traits like the obesity, diabetes, like heart, like blood pressure. So there are many GYs. NIH supported thousands of projects, generated several thousands of genome-wide assertion study, large scale. And recently we first developed a tissue-specific enrichment analysis to show those gene expression regarding tissue specificity. So tissue specificity we can see those can enrich the tissue for the genetic association signals from like GYs. It's a large scale. We collected all the available genome-wide association study, more than 5,000. And today, I mainly focus on the second part, is the natural extension. We want to see whether those kind of genetic variants from association study can see, we can see the cell type specific enrichment. So this work is mainly from two junior faculty, Guangxing Pei and Yulin Dai, and also another colleague, Pei-Hing Jia here. So to talk about cell type specific enrichment, I talk first is because we use the same data set called the tissue specific enrichment analysis. First, we collect all those kind of GWAS data set. As I mentioned, it's a large scale, more than 5,000. Even UKBB, UK Biobank has more than 5,000 data sets. So we put them together, do quality control, and then calculate, gene-based p-value for the association study. And then we have a list of genes. Our purpose is to detect, for any gene, whether this tissue with a specific enrichment for those genes or a set of genes. So we develop a method called the DETS, and also then do the gene set enrichment analysis. So then, because our purpose today is mainly cell type, so we look at the cell type specific enrichment, and this is a more, simple view. You have brain, and the brain has different subregion, and each subregion has different, cell type. The expression by the cell type, for example, the bronchotic, you have EX1, excitatory, kind of cell type. You have many kind of cell types. Then what we do is we develop this pipeline. First, as I mentioned, we collect many thousands of GWAS data set, and then process them, get each of the gene associated with the GWAS specific disease or phenotype, and then we collect the right side, the single cell data set, mainly from like a human cell atlas, human cell landscape, and some other data sets. And then we filter the cell type, the genes without, this high enough level expression. And then we use, those kind of cell type specific enrichment analysis. Mainly it's a statistical test. And then we put them together to develop a database. So as summary, this database has more than 5,000 GWAS data set. And also we collect more than 900,000 cells from leading those kind of single cell, transcriptional consortium and the literature. And this covers more than 550 tissue cell types from 71 adults and fetal tissues. And then we do this analysis. So our goal basically, for each pair treat tissue cell type, we can, show this kind of relationship. Like in this case, we use the pancreas as an example to show how those kind of tissue and the cell type related based on the gene, for those kind of, the gene expression and the genetic association. And finally, I just, remind again, our call is available to the investigators, and we try to do our service as much as possible during the COVID-19 pandemic. And finally, I also like to thank the team here, those people I mentioned for the projects I mentioned today.
Okay, thanks, Nguyen.
Okay.
So yeah, it seems that we take a longer time than we thought. It's the first time. Sorry for that. But I think we're happy to open the floor for any question. You can either type into the chat box or you can just unmute yourself.
Yeah, hi, Ray. There's already a lot of questions in there. Should we maybe pick some and we can discuss them a little bit?
Sure. I see there's a single cell RNA-seq and a nuclei, single cell nuclei.
Yeah, there's a bunch of questions related to should you do single nucleus RNA sequencing or single cell RNA sequencing? So maybe we could talk about that a little bit. Seems to be a few people interested.
Yeah, go ahead.
Oh, sure. Okay. Well, so single cell RNA sequencing, the limitation is that you need either fresh cells that you dissociate from tissues pretty quickly after you obtain them, which is not a problem for mice really, but can be a problem for surgical samples. You can cryo store those cells in freezing media like DMSO, and then wake them up later to run the experiments. So that's a possibility. But you can't really analyze frozen tissues, because as soon as you freeze the tissue, the plasma membrane gets ruptured by the ice crystals, and so the messenger RNA just leaks everywhere. So from a frozen sample, you can't get intact cells unless they're in the media. So that's where single nucleus RNA becomes quite useful. A lot of people now are using it for frozen tissues. So in a frozen tissue, the nuclear membrane stays intact through freeze-thaw cycles, and you can isolate nuclei from a frozen tissue and run a single nucleus RNA sequencing assay. But the difference is that single nucleus RNA is very different than cellular messenger RNA. So if you're trying to look for the top markers that you often would know for a cell type, they're going to be very different in the nuclear RNA. So it really depends what you're trying to look for. Single nucleus RNA is also useful for some very difficult cell types. So I'm sure Ray has a lot of experience with this, in the retina, but other ones are like neurons, for example. The nucleus is nice, small, and round, but the neuron is very large. And adipocytes is one that we deal with in the breast. The cells are gigantic, 300 microns, but the nucleus is very small. And so if you isolate nuclei first, you can profile things like adipocytes. So those are some of the advantages, disadvantages. I mean, if you have access to fresh tissue or viable cells, I think it's always better to run the single cell RNA sequencing. But if all you have is snap frozen tissue or OCT, frozen tissue, you can still think about running the single nucleus RNA sequencing assays. So yeah, I don't know, Ray, do you have any other things to add to that, or Jiangming? I noticed you said you were running that as well.
I think it's pretty comprehensive. You said that. I think a lot of time, if you have fresh tissue, if you can isolate the cells and with good viability, I think the single cell RNA-seq seems, I will try that single cell RNA-seq first. With caveat that you may have a sampling bias. Some of the cell type may be undersample or oversample when you do the single cell RNA-seq. But a lot of time when we work with different investigators, some tissue type like brain, it's hard to really isolate clean viable cells without a lot of debris. So in those cases, you may be better off just go straight to the freeze it up and get the nuclei out of it. So, Zhongming, do you have anything?
Yeah, I'd like to add a little bit more, this argument about, how many genes can be reliably detected in single nucleus RNA sequencing versus fresh tissue. Basically, some people believe most of them can be covered. Some people still argue fewer. That's one. Second is which kind of gene depends on, very important. Your research, this is very important for your specific research, because some gene, for example, for the brain research, you maybe, I heard about some people talk about this. I haven't spent much more time. You have to be careful because those genes may be, more affected in the can be more, maybe, for those genes, the expression is different in cytoplasm versus nucleus. And you only see those gene expression changes in nucleus, not in cytoplasm. But when you draw the conclusion, you always think, this is my conclusion in gene expression difference in this specific research area. So that has to be very careful. There's some limitation because it's more restricted in terms of the gene expression in the nucleus only, not in the cytoplasm. Yeah.
So yeah, there's this question on whether the blood cells. So actually, blood cells or immune cells can be quite easily, even for human, can be easily quite preserved. And then when you store them, they still have very high viability. So for blood cell, actually, it's most easy sample to do anything on them, right? So they're really robust. So I think you can design based on your experiment. Because a lot of time, when you do immune profiling, you also want to know the surface marker, the protein. So in those cases, you need to start with the single cell instead of nuclei. You want to profile the surface CD, CD4, CD8 markers. Another thing I see from the chat is, typically in our experience, in terms of the cell type, the single cell RNA-seq frequently have drop off of some cell types. Single nuclei is less prone to that issue.
So yeah, there's a question about will single nucleus RNA-seq result in less cell types? In our experience, it actually results in more cell types. I think that's what Ray is saying too. Like when we ran it on breast tissues, we got all the same cell types as the cells, but we also got adipocytes, we got mast cells, we got some apocrine cells, so other cell types that are more sensitive to anoikis and dissociation and kind of aren't present in your cell populations. You can see in the single nucleus RNA-seq data. But in my opinion, I mean, I think the single nucleus RNA sequencing data is kind of, Ray might not like this. I think it's kind of garbage because, you get all these genes and you can't really use them for pathway analysis or signature analysis. They don't make sense in terms of cell type identities, at least, for stuff that we see in the breast and, colon and other tissue types. So I really think if you can, try to do cell analysis. If there's certain cell types that you can't get, like adipocytes, you don't have a choice, then, do nuclei analysis. I mean, the only other thing that it's useful for, I think, is looking at the frequencies of the cell types, because the single nucleus data, you very quickly dissociate the tissue into a nuclear suspension. So the cell type frequencies are more true to the original tissue. But they're still distorted, I'm sure that's even with nuclei, not everything is gonna dissociate at exactly the same rate. So that's one other advantage. But in terms of the actual genes you measure, if you want to, if you look at T cells, you're not going to see CD4 or CD8, or any of the typical markers, FOXP2, anything like that. They don't show up in the nuclei data. You have all these other genes that just aren't typical, genes that people, an immunologist would recognize. So yeah, so therein lies the problem a little bit.
So I see a question from Sung Zhang about other species. Yeah, so single cell can be applied to pretty much all the species. At least animal species and also plants too. So in our core, we definitely have done Drosophila. We've done a lot of Drosophila, and also C. elegans, human mouse. So as long as you can get enough good live isolated cells, you can do the profile. So I also see the multi-omics from TJ. Yeah, essentially this is just like you do ATAC and RNA-seq in the same cell, but sort of in parallel. Experimental-wise, you make two distinct library and sequence them. So the sequence depths is comparable as you do, individually, right? So it's two independent library you make. Does that answer the question? And also how to multiplexing. Yeah, you can do cell hashing, that sort of thing with multiplexing.
Are you sure about that, right? I don't know any multiplexing methods for the co-assays.
It is a nuclei lipid-based antibody. I think the next thing that they will release support that pretty soon. But right now you can just buy the lipid-based antibody hash.
The multi-seq?
Yeah.
Oh, okay, I haven't seen it commercially available.
Yeah, it's not commercially available, but you could do it. Yeah, it's biology have the reagent.
Oh, okay.
But we don't have tons of experience ourselves on multiplexing. I don't know about Nick and Zhongming. Do you have done a lot of multiplexing yourself?
I don't, but we do some computational way, basically going back and forth, right? The bulk tissue to single cell to bulk tissue kind of a comparison, but we haven't done for multiplexing for single cell RNA sequencing.
Nick, have you guys use?
Yeah, we have our own method that we developed. It's we just submitted it for publication, so it hasn't been published yet, but it works pretty well internally for RNA and for ATAC experiments. But we don't have that much experience also with cell hashing. We've done some site-seq, which is very similar, but we haven't actually done not a lot of people have requested doing the multiplexing yet. So other than our own technique, we haven't used the other commercially available methods yet.
Yeah, we based on our experience, you definitely can do multiplexing. But also people, I mean, there's two purposes for multiplexing, right? One, well, there's multiple benefit. One is that you can identify doublet more accurately because you have two cells have different barcode, you know they are from two cells. And the second thing is if your sample, you don't need that many cells. You don't need 10,000, for example. You want 3,000 cells per sample, but you need to cover many samples. You can multiplexing so that you can reduce the cost. The third one, people thinking about to jam it, so we increase number of cells from the machine. We tried that one. That doesn't work well for us. It's not supported by 10x, and then actually it leads higher failure rate when you try to put a jam. So and the clogging issue. So I think that one is sort of risky. It's possible, but I don't think there's a huge benefit. So let's see. Is any Zhongming, you have a question for you. How does the DrivAER ensure that we detect the useful feature rather than noise in the bottleneck of the AutoEncoder?
Okay, yeah, I saw this now. So I honestly, I'm not very sure this answer can be quick, accurately answered. But in general, I think the AutoEncoder, basically we do encoding, decoding, and it's a feature hidden feature discovery, and these kind of things we do. It's hard to know how this bottleneck can change later on as a noise. But the second part, we have the machine learning, like the random forest. We also tried several other machine learning techniques, and we compare which machine learning can be better to identify those informative transcriptional programs. And we demonstrate in both the real data set and the simulation data set, and compare to the peer methods to demonstrate that this indeed is a more informative. And this probably partially answer your question, but the AutoEncoder part, I guess it's very difficult to say which one is noise, which one is the true features. If we can probably we can even do the first part. I wish Lucas is here. He is the main person for this technique. Yeah, I will bring this question to Lucas and answer you. I saw your name, Saifu. If you send me email, probably we can help you to figure out. Okay.
So,
question here, actually, curious to see what you guys think about it too. Is kind of about what does ATAC-Sea bring in addition to single cell RNA sequencing? Or the co-assay versus just single cell RNA sequencing, especially if you're only interested in cell types. I mean, of course, you can, in my opinion, I think the ATAC-Sea really doesn't bring that much more onto the single cell RNA sequencing. Now, of course, you can integrate the two data types and try to define cell types better. And you could potentially look at gene regulation in a lot more detail if you're interested in identifying transcription factors that regulate cell states. But for just defining a cell state or a cell type, single cell RNA sequencing itself, I think, works pretty well. I don't know, Ray, do you have some opinions on this as well?
So I think, in terms of the cell cluster, single cell RNA-seq is performed definitely much more informative than single cell ATAC, for sure. So the single cell ATAC, I think, is add on the gene regulatory region. If you're really interested in how the gene transcription is regulated, and the ATAC-Sea is the way you can get to those regions, and looking for that. Another thing I think, in my opinion, is useful is the ATAC-Sea give you the DNA sequence. So it's also possible to link with the GWAS data, because there's a variation you can sample from the DNA sequence. So I think those are two major usage we are used, basically looking for gene vector region or connect with GWAS.
Yeah, there's another question here about, is it possible to infer spatial information from single cell sequencing data? In general, the answer is no, because the first step is you dissociate all the cells, and so you inherently lose all the positional information of the cells in the tissue. But there are other platforms like spatial transcriptomics and a whole series of in situ sequencing methods that can do that. However, I've seen one or two papers where they try to do a computational reconstruction of where cell types might be located in the tissue. But I'm not sure how accurate those methods really are. I think the general rule is that, you know, you basically sacrifice all your spatial information as you initially dissociate the tissue.
Yeah.
I don't know if unless I don't know, Jiangming, do you know any techniques to do spatial reconstruction from just single cell RNA data alone?
Yeah, people typically think about this, you know, the temporal way, not the spatial way. Obviously, from computational side, as long as you see the connectivity, right, from one spatial region to another spatial region, you can always make connection. But how reliable? Yeah, I'm not very sure. So as you mentioned, the spatial, the you know, like single cell 10x Genomics has just provides the new platform. So I think at this stage, probably, you know, more likely we generate data from such kind of experimental design, and then allows us to develop a computational program to see how reliable we can infer such kind of feature using the real data and the simulation data. But to my knowledge, you know, in the field, we are still more using temporal way, more trajectory way, like, you know, the lung injury example I demonstrated to show the relationship. That's more reliable.
Yeah, I mean, I just want to add there are multiple method or technology have been developed for spatial single cell technology, like the SLISEq and MERFISH, SIGFISH, and then also the commercial one, Visium. They are not true single cell, but they have a cluster of 10, 20 cells. So, but generally, because if the single cell RNA-seq you already dissociated, so as Nick said, the spatial information is gone, and then you just try to infer them back one way or the other, not necessarily to be reliable. So we are planning to have a panel discussion focused on spatial single cell genomics later or mid next year.
Yeah, but I think the question is more like if you have a single cell RNA sequencing data, can you infer the spatial arrangement?
Yeah. So there were one or two papers on it, you know, but they were in highly differentiated tissue types where there's like very clear structure. So how generally applicable it is, I'm not sure. Yeah. I think for now, you know, in general, if you're dissociating a tissue, you're kind of sacrificing the spatial information. Although there's a lot of great technologies, you know, that are becoming available to profile cells or small regions, you know, across space, and for, you know, that in combination with single cell RNA sequencing can be very powerful, because you can know from the single cell data, these are all the cell types and cell states, and they can use the spatial data to map them and understand how they're organized, who are their neighbors, and how are they interacting.
So do you have more? We do have many questions, but I think the timing is already 10 minutes after. So do you want to go on to continue answer?
We still have 89 people there here. So
yeah.
maybe we spend
should we do one last question? The last one here, it's kind of interesting. For detecting rare cell types, what are the general factors one should consider in terms of experimental design? That's a good one. It depends how rare they are. You know, you're talking 1 in 100, 1 in 1,000, 1 in a million, because you have to go through different processes to get rarer and rarer cells. If you're talking about 1 in a million, you need, you know, basically platforms to isolate them, like CTC platforms. There are a whole bunch available. We like RareSite, for example. And there the markers are really important, because you're trying to label those cells in some way and then image them and then isolate them with a robotic micromanipulator. So, so the considerations there are mostly what are the positive or negative markers that you're going to stain with. and whether you need DNA or RNA, because if you need DNA, you can do all the fixation steps and do imaging. If you need RNA, it's really, really difficult. If you're talking about more like 1 in 1,000 or 1 in 10,000, you can use enrichment strategies, you know, like FACS or max columns, and try to get them up so that they're around 1% or 5%, and then you can run your experiment. You can also try to purify them completely with FACS if they're, you know, not that rare. If you don't know the markers well, then it becomes more difficult because it's unbiased. But yeah, it's that's probably one of the harder areas, I think, of single cell genomics right now. And most of, I'd say the most advances in that area have been in the around circulating tumor cells.
I think, you know, I just want to sort of add a little bit on Nick, you know, especially if I try to understand, you know, where you're coming from, the question from me is, if you just say 10x RNA-seq, the detection, you know, if you don't know too much, just go in unbiasedly. I would say if you generate about 8,000, 10,000 cells, you can find cell population around like 1%-ish. You'll be able to find them, and then particularly they form relatively distinct, you know, profile. So sort of detection sensitivity, maybe that's what you're trying to. If yours is 0.1% or less than that, then some sort of enrichment is needed before you put them in. Otherwise, you'll not be able to find it.
All right, we end it there.
Yeah, I think it's because of the time. Sorry, we need to cut it short. I mean, I'll not be able to finish all the questions, I guess. So we'll be recorded, and if you have more questions, feel free to email me and Nick and Zhongming. Right, we're happy to help out. Thank you very much.
Bye, everyone. Have a good afternoon.
Okay,
Thank
bye-bye.
you.